Hello, everybody. Welcome one and all to the Kalim Red Talk series. I am Bertrand Edel, director of the Kalim Institute, your host for the Red Talks. As you may know by now, the Red Talks are webinars for lymphomaniacs. It's every first Thursday of the month, 6 p.m. CET, open access, free, just register as you did today, that's it. By now, you all know how webinars work. You have a question tabs on your right, you have a chat tab on your right, you can participate on both tabs. After the webinar, the Q&A session will remain open for an extra half an hour. So about 7:30 till 7:30, you can keep asking questions, and we'll try to have as much question answered by the end of this seminar. So I have an announcement to do today. Our guest star, Ari Melnick, had unfortunately a last-minute personal issue and finally cannot come and join us. So he asked me to apologize to all of you. We're very sad and we miss you, Ari. That's going to be for another time. But the good news is that he didn't want to let us down, and Wendy Beglin kindly accepted to deliver the Red Talk lecture for us. So many thanks, Wendy, for this last-minute swap. Looking forward for your talk, but I let our other guest stars, Laura Pasqualucci and Frank Marschauser, introduce the Red Talk. The floor is yours, Laura.
Thank you, Bertrand, and good afternoon, everybody, or good evening, depending on where you are. I'm very pleased, actually, to have the opportunity to introduce Dr. Wendy Beglin from Weill Cornell Medical College in New York, who's stepping in on behalf of Dr. Melnick. Dr. Beglin obtained her PhD degree in biological sciences at the University of Buenos Aires in Argentina, where she first developed her interests in the area of epigenetic control and transcriptional regulation before moving to Cornell for her postdoctoral training in the laboratory of Ari Melnick on the field of hematology oncology. And there, I believe she was really instrumental in pushing forward the field of epigenetics in lymphomas. In fact, I think lucky Ari to have her, as she contributed to the identification and functional characterization of novel epigenetic and transcriptional mechanisms that contribute to humoral immunity, as well as to the normal germinal center B-cell differentiation process and to the malignant transformation of these cells, with a very special interest in the polycomb-protein. And this, I think, is an interest that she has continued and will continue to follow now as an independent investigator after joining the faculty of Weill Cornell 3 years ago, where she's currently an assistant professor of pharmacology and medicine, and where I believe she's going to be fully committed to bring new ideas toward the understanding of the role of epigenetic perturbations, and in particular EZH2 mutations, also trying to translate these findings into the development of novel therapeutic principles for these malignancies. So as I mentioned, I think Wendy has provided some significant contributions to the field. She has an impressive track record of publications, but in particular, she led a number of seminal studies on the discovery first and then functional understanding of the polycomb proteins and EZH2 in particular, this methyltransferase that is commonly mutated in both follicular lymphoma and diffuse large B-cell lymphoma. So she went all the way on from the discovery of the role of this protein in the germinal center reaction to the consequences of point mutations that we find in patients, and now more recently exploring a novel area whereby she's trying to understand how these alterations can instruct the surrounding microenvironment and the germinal center accessory cells. So I think if you want to write a review on this topic, you will almost only cite Wendy's papers. And for this work, she has already earned a number of awards. She has a Scholar Award and more recently a Young Investigator Award from the American Society of Hematology to continue pursuing these studies. And today, I think she's gonna be speaking to us about all of this. Thank you very much, Wendy, for joining us.
Thank you so much, Laura. Thank you for your kind introduction. And thank you for inviting me on behalf of Ari. I'm sorry that he couldn't join today, but let's see what if I can do, if I can convince you about the actions of mutant DCH2. I'm going to share the screen. Okay. Can you see my presentation in full screen?
Absolutely. That's great.
Okay, I can't see you. I just see the presentation, but okay. So first of all, I'll give just a brief introduction about the epigenetics in germinal center reaction and lymphomagenesis, and then I present our work about specifically how mutation in EZH2 cause lymphomas. So, as you know, germinal center B cells are actually a complex mixture of cells undergoing several transitional states. In the germinal center dark zone, centroblasts undergo massive proliferation and clonal expansion, while at the same time they are mutating their immunoglobulin genes through the process of somatic epimutation. Once the centroblasts have stopped proliferating, they are known now as centrocytes, which are located in the light zone of the germinal centers, and they are subjected to selection by follicular helper T cells. And this happens in the presence of follicular dendritic cells. The negatively selected centrocytes undergo apoptosis, and the positively selected centrocytes may recycle to centroblasts and re-enter the dark zone to get more additional cycles of somatic happy mutations and keep dividing. And only the few B cells that were able to develop mutated immunoglobulin genes with high affinity for antigens are now allowed to survive and exit the germinal center as differentiated plasma cells or durable memory B cells. Now, how do B cells are transiently deviating from their this program differentiation path to form the germinal centers? For those B cells that will form the germinal center, the B cell differentiation program has to be silenced. So cell cycle inhibitors and also DNA damage checkpoint genes must be silenced to allow this high proliferative germinal center phenotype. And all these dynamic phenotypic changes are actually epigenetically controlled. And this function is in part mediated by the histone-methyltransferase EZH2, which is highly induced in germinal center B cells, and it's required for germinal centers to form, for the B cells actually to form these germinal centers. And indeed, as you see on the top right, the conditional deletion of EZH2 at the pre-germinal center stage prevented the germinal center formation. And these switches that turn on and off the epigenetic programs are actually very important because these germinal center B cells give rise to the great majority of B-cell lymphomas, including follicular lymphomas and diffuse large B-cell lymphomas. So somatic mutations and epigenetic modifiers, both in epigenetic modifiers actually and transcription factors, are a hallmark in the germinal center derived lymphomas, both FL and DLBCL. We know that the somatic mutations induce irreversible cancer phenotypic hallmarks. Now, germinal center B cells manifest many of these cancer hallmarks, such as sustained proliferation, genome stability, and mutagenesis, but this is in a transient and reversible manner. Most mutation in epigenetic modifiers in both FL and DLBCL result in gain of repression or loss of activation mechanism. And founder mutations in germinal center-derived lymphomas have the function of maintaining the GC phenotype. And as I mentioned before, during the germinal center reaction, there are many vulnerable transitional states that can give rise to lymphomas. So how the epigenome control gene promoters and enhancer? Well, there is an equilibrium between histone writers and erasers that maintain the germinal center epigenome in a dynamic and also reversible state. And this equilibrium is destroyed by somatic mutations in epigenetic modifier genes, such as the methyltransferases EZH2 and KMT2D, and also mutations in histone acetyltransferases like CREBP and P300. So going back to our germinal center cartoon, some common epigenetic somatic mutations in lymphoma, what they do is to mediate their effect by disrupting the plasticity of these immune synapses. I come back to this later. So now how mutations in EZH2 cause lymphomas. EZH2 is a core component of the polycomb repressive complex 2, and it methylates lysine 27 of histone 3. This is a histone mark that is associated with gene repression. And we found that what EZH2 does at promoters of germinal center B cells is to form bivalent chromatin by trimethylation, the lysine 27, in histones that were previously marked in the naive B cells by K4 trimethylation. K4 trimethylation is a mark associated to activation of transcription. So as a result, EZH2, when it's expressed in the germinal center, it transiently posts the expression of genes that are involved in cell cycle checkpoints like P21, P14, P16, and also genes that are related to exit of the germinal center. For example, here are two germinal center exit genes, RF4 and PRD11, and you can see that they are marked by K4 trimethylation in naive B cells, and they gain K27 trimethylation in the germinal center stage. And we have actually proved that H3 K27 trimethylation on one specific gene can mediate the EZH2 germinal center phenotype. This is the case of CDKN1A, P21, which is highly enriched in H3 K27 trimethylation, and as you see on the top right. And what we showed is that when we knock out CDKN1A, it rescues the EZH2 deficient germinal center phenotype. And in this work, we found a positive feedback loop in which EZH2 controls germinal center B cell proliferation by suppressing P21, and this enables cell cycle progression. And so this also induces the phosphorylation of RB and release of the transcription factor E2F1. And E2F1 positively actually regulates the expression of EZH2. So how are the germinal center bivalent promoters repressed? How does EZH2 mediate cell context dependent functions specifically in the germinal center B cells? We found that in germinal center B cells, both EZH2 and BCL6, the transcription repressor BCL6, cooperate to recruit a non-canonical PRC1 B-core complex. So this PRC1, non-canonical PRC1, contains CBX8, And in this manner, it represses the transcription of these bivalent genes. So CBX8 binds to bivalent promoters, and this binding enables the specificity for the recruitment of PRC1 BCOR. So the recruitment of EZH2 and BCL6 to chromatin is actually independent of each other, but they are both required, dual key mechanism to tether the non-canonical PRC1 BCOR complex and then launch the germinal center reaction. Up to 30% of patients with the GCB subtype of diffuse large B-cell lymphomas and the follicular lymphomas have gain-of-function mutations in EZH2. Mutant EZH2 drives actually an aberrantly efficient trimethylation of lysine 27 in histone 3. And the wild type is much more efficient to catalyze the monomethylation of H3K27. So to study the impact of these lymphoma-associated gain-of-function EZH2 mutations in vivo, we generated the mutant EZH2 conditional knock-in mouse model, where the point mutation Y641F was introduced in the endogenous EZH2 locus. So when CRE is present, mutant EZH2 is expressed. And we cross this to sigma-1 CRE mice to induce the expression of mutant EZH2 specifically in germinal center B cells, because sigma-1 encodes for IgG1, which is induced when B cells get activated. So as expected, germinal center B cells, but not naive B cells, showed higher levels of H3K27 trimethylation, and I am not showing here, but the levels of EZH2 were not changed. And the mutant EZH2 mice developed germinal center hyperplasia, as shown here by flow cytometry, where we used the germinal center markers FAST and GL7, and also by immunohistochemistry, we used here the germinal center marker PNA. Now, the germinal center expansion was observed only in heterozygous mutant EZH2 mice. The homozygous mice failed to form germinal center. And this is actually consistent with the fact that, as I told you before, the wild-type EZH2 is more efficient in mediating mono and dimethylation, and the mutant is more active at converting di to trimethylation. So mutant EZH2 requires the cooperation with wild-type allele. And this also explains why the gain-of-function mutation of EZH2 in patients is always in heterozygosis. When the gain-of-function mutant EZH2 is combined with frequently mutated oncogenes that occurs in B-cell lymphomas, such as BCL2 on the left or BCL6 on the right, it accelerates lymphomagenesis. So with the EZH2 model, we decided to explore the mechanisms through which EZH2 mutations disrupt the immune system to initiate malignant transformation. Okay, so for most of our experiments, we did a mixed chimera bone marrow transplantation. So in this context, wild-type cells are directly competing with the mutant EZH2 in the same microenvironment. So we mixed here 50% of CA45.1 wild-type, shown in white, and 50% CA45.2 mutant bone marrow, shown in blue. And I'll keep white for wild-type and blue for mutant EZH2 throughout the presentation. So we injected the mice, the bone marrow from these mice in the RAC1 knockout host mice that don't produce mature T or B cells. And the recipient mice were then immunized with the ship blood cells to induce germinal center formation and euthanized days later, which is the peak of the germinal center reaction. And we found that mutant EZH2 provides an advantage to activated the B cells to seeding the germinal center reaction. And when the initial ratio of the cells in the chimera was skewed towards the wild type, this difference was even higher. These are immunofluorescence pictures of a sphenical germinal center, where you can see. So there is a higher overlap of the germinal center marker PNA, which is here in green, with CD45.2, which is the mutant DCH2 in purple. And this overlap is higher than the wild type EZH2 in red. So this shows that the mutation is providing a fitness advantage to germinal center B cells. So then we evaluated the proportions of centroless and centrocytes by flow cytometry using very well-established markers, CXCR4, CD86, and CD83. And we find an expansion of centrocytes in mutant EZH2 mice. And using the YFP reporter mice, we confirm that this expansion of the light zone occurs in the mutant EZH2 germinal centers. These are pictures of spleen sections, and we stain the follicular dendritic cells with anti-CD35 antibody, which is here shown in blue, and this is to highlight the light zone. And you can clearly see the expansion of the follicular dendritic cell network in the mutant germinal centers here on the bottom. We also confirmed this by, this is a mass cytometry imaging using the site of Hyperion. And in these spleen sections, the follicular dendritic cells are shown in green. And you can also appreciate on the right the increased levels of H3 K27 trimethylation in the mutant EZH2 germinal centers. Okay, because the EZH2 inhibitors cause primary induction of the plasma cell differentiation and also proliferation arrest in cell lines, we thought that the light zone expansion would be caused by failure to exit the germinal center reaction. But we were wrong. This was not actually the case. When mutant EZH2 mice were immunized with specific antigen NP, they produced normal levels of NP positive memory and plasma cells. This was 12 days after immunization. And they also produced the bone marrow NP positive long-lived plasma cells when we tested this is 76 days post-immunization. So the anti-NP specific IgG production was also normal in mutant EZH2 mice that we tested here by ELISpot and ELISA assays. And we confirmed this normal differentiation to plasma cells using the YFP reporter Cree mice that were crossed with the C-gamma1 Cree. So the bright YFP cells outside the follicles are plasma cells. They could also be extra follicular plasma cells. And we also stained them with the CD138 in red, that is here in the magnified insert. So next, to evaluate the advantage of the, if this advantage of the mutant could be explained by proliferation. So it's not the germinal center exit, but maybe they are actually proliferating more. And indeed, we observed aberrant centrocyte proliferation, here identified by Ki67 staining in the chimera mice. But to more precisely evaluate if mutant TCH2 centrocytes are truly proliferating, because normally they don't proliferate, we use the Fuji strain that encodes probes that fluoresce red during G1 and green during SG2M phases. So, early S would be yellow, and late S, G2, and M would be green. So this model allowed the study of this the cell cycle dynamics. And you can clearly see here by flow cytometry and also immunofluorescence microscopy that the resting naive B cells are mostly red, M cherry positive, while germinal center B cells are more either double negative or green. This is Venus positive. So we crossed the Fuji mice with the wild type and mutant TCH2, we immunized them, and we found a higher proportion of mutant TCH2 centrocytes in SG2M and lower number of cells in G1 phase. So this indicates that there is like an aberrant proliferation of mutant TCH2 in the light zone. And the centroblast didn't show big changes. These are confocal microscopy images of the mutant DCH2 Fuji spleens on the right that show green proliferating cells in the light zone, which is identified again by CD35 staining in blue, while in the wild type DCH2 germinal centers on the left, these green cells are most restricted to the dark zone. So we next evaluated apoptosis to see if this expansion was also explained by this mechanism, and we observed that the light zone expansion is partially also explained by reduced apoptosis of the mutant TCH2 germinal center centrocytes. So far, this data shows that EZH2 mutations is driving germinal center hyperplasia through enhanced proliferation and survival of centrocytes. But as I said before, given that the germinal center B cells are actually a complex mixture of different subpopulations undergoing several transitional states, we decided to study the transcriptome of these cells at the single cell level. So we sorted the YFP positive and the IgD negative cells in the YFP reporter mice. So these cells will include germinal center B cells, memory B, and the plasma cells. And then we did the droplet-based single-cell RNA-seq using chromium 10x. And so focusing on the germinal center B cell population, I'm not showing here the initial plot where we had the plasma cells. This is just focusing on the germinal center B cells. We identified 8 clusters based here, what I'm showing here on the right, the expression of some gene sets, and also based on expression of individual hallmark genes. So we found, as expected, the expansion in centrocytes in the light zone. And unexpectedly, we found the significant reduction of centrocytes expressing the MYC-associated recycling signature. So this suggests that mutant TCH2 is impairing recycling to the dark zone. But to confirm the reduction of these recycling cells, and also because these cells can be identified by MYC expression, we crossed our EZH2 mice with animal expressing the GFP MYC fusion protein from the MYC locus. And we found a reduction of GFP positive germinal center B cells in mutant EZH2 mice. So this indicates that there is a failure to re-enter the dark zone in the case of the mutant EZH2 centrocytes. But next, we went into the mechanism of action, what actually EZH2 is doing, the trimethylation, and to evaluate the effect of the mutation in the histone landscape, we did the first histone mass spec in germinal center B cells, and we found, as expected, a massive increase in H3 ketone trimethylation in the mutant EZH2 cells. And this was at the expense of a reduction of the dimethylation. And this is interesting. We also observed an increase of unmethylated H3 K27 in mutant cells at the expense of a reduction of in the monomethylation. And this could be explained because EZH2 point mutation is actually loss of function for monomethyl activity, as I explained before. These are ChIP-seq experiments that show that while the majority of promoters, H3 ketone trimethylated, were shared between wild type and mutant, there was also gain of 2,800 de novo promoter peaks in the mutant EZH2 cells. And these de novo H3 ketone trimethyl promoters in mutant germinal center B cells tended to be close to promoters that were already trimethylated in the wild type cells. So this suggests that H3 ketone trimethylation is spreading from nearby promoters. Then we also did the RNA-seq, and the analysis identified 650 genes that were downregulated and 270 genes upregulated in mutant EZH2 germinal center B cells. And the repressed genes showed H3 ketone trimethylation peak at the transcription start sites as expected, and this spreading of H3 ketone trimethylation both up and downstream. The upregulated genes on the right did not contain any defined ketone trimethylation peaks at the transcription start site. So to understand how transcriptional programming is affected by mutant EZH2, what we did was to calculate the trajectory of gene regulation from naive B cells to centroblasts and then centrocytes in mutant EZH2, and we compared this to trajectory in the wild type cells. And then we grouped genes based on their pattern of perturbation. So for example, in the cluster that I'm showing here, these genes are upregulated in the wild-type centrocytes and fail to upregulate, I mean, they are kept repressed in the mutant EZH2 centrocytes. So by doing this analysis, we identified 8 distinct gene expression modules, which are based on the trajectory of genes across these cell types. And among genes in the modules 5, 6, and 7, which are genes that fail to de-repress in the mutant centrocytes that are kept repressed, we found genes involved in the immune synapse with T follicular helper cells. And actually, some of these genes showed also higher levels of ketone trimethyl in their promoters. Here, I'm showing the ChIP tracks for 2 examples, CD69 and ICAM1. So this suggests that they are direct targets of EZH2-mediated repression. And among upregulated genes in the mutant centrocytes in modules 1 and 8, we found genes that were involved in interactions with follicular dendritic cells, such as here, BAF receptor, lymphotoxin, and TNF. So in the germinal center light zone, B cells require these strong interactions with T cells to survive and to be selected for differentiation. And the RNA-seq data suggests that this process might be impaired in the mutant TCH2 centrocytes. At the protein level, by flow cytometry, we also found that many of these proteins involved in BET immune synapse were downregulated in mutant centrocytes. But then we went on to evaluate if centrocytes that were loaded with OVA antigen could preferentially interact with OVA-primed OT2 TFH cells. These were, as I said, were carried out ex vivo. So we did this by flow cytometry, and we assessed the formation of the BET conjugates by gating on the duplets. And we found that there were less conjugates in the mutant DCH2 cells. So this suggests that the mutation is impairing the formation of these stable interactions between centrocytes and TFH. And then in an in vivo assay, what we did was to block the interaction between GCB cells and TFH by using an anti-CD40 ligand antibody. And we found that, well, the treatment of a control wild-type mice with this anti-CD40 ligand antibody inhibited the germinal center reaction as expected, but it didn't significantly affect mutant TCH2 centrocytes. I mean the germinal center formation. And in a chimera experiment, the mutant TCH2 germinal center B cells had, as I showed before, a competitive advantage over wild type in expanding the germinal center. And this advantage was even greater when we blocked BT interactions. So this suggests that EZH2 mutations make germinal center B cells less dependent on T cell selection. And these results were then validated by blocking the interactions between B and T using an anti-ICAM1 antibody. Now, the T cell help is required from centrocytes to recycle back to the dark zone for additional mutations. So this is actually directing the clonal evolution of the immunoglobulin loci. So to the evidence of this differential selection pressure in the mutant DCH2 light zone prompted us to evaluate the clonality of these cells. So for this, what we did was to cross the mutant DCH2 mice with the stochastic multicolor Cree recombinase reporter confetti. And when you use them in homozygosis, there are 10 different possible combination of colors. So this model allows a way to label and distinguish individual germinal center B cell clones when we cross the mice to C-gamma1 Cree. A polyclonal germinal center will feature many different color combinations, as shown here on the left-hand side, and the dominant clonal germinal center will show few combinations, as you can see here on the right. We found that mutant DCH2 germinal centers showed less combination of colors in their germinal centers. So this is that they established a clonal dominant pattern in contrast to the wild type situation. But we still need a definite genetic proof to claim clonal dominance. So what we did was single cell genomic clonality assays, and where we sequence IgM and IgG variable regions from individual germinal centers. So for this, we used both YFP and confetti reporter mice. And we found that the mutation frequency of wild-type IgG1 was significantly higher than the mutant DCH2 cells. And we observed a reduced diversity of clones in the EZH2 mutant mice. If you look at the phylogeny trees of the clone, the centroblasts, the branches of the centroblast, which are shown in orange, and the centrocytes in purple, and you can see that there is like a clonal dominance in the light zone of the mutant EZH2 clones because of the number of branches. So altogether, these results show that EZH2 mutant fail to engage T cells, so they are not receiving proper TFH signals that would induce recycling to the dark zone. And in contrast to the normal cells, which cannot survive without T cell help, the mutant centrocyte can even proliferate in the light zone. So which cells are supporting the EZH2 mutant centrocyte survival? We postulated that they might be instead dependent on follicular dendritic cells, especially because of the expanded FDC networks that we observed in the pictures I showed before, and because of the upregulation of genes involved in interaction with FDC signaling that we found by our RNA-seq analysis. And indeed, we found that BAF receptor, lymphotoxin beta, and TNF transcript were upregulated in EZH2 mutant centrocytes. And at the protein level, by flow cytometry, we confirmed BAF receptor upregulation in centrocytes. So as the maintenance of FDC network and the germinal center required signaling through lymphotoxin beta. So to determine if EZH2 mutant germinal centers were dependent on these FDC's interactions, what we did was to treat the chimera mice with soluble lymphotoxin beta receptor. And as expected, we found a severe reduction in the abundance of germinal center B cells. But EZH2 mutant germinal center B cells appear even more sensitive to FDC blockade when we compared to wild type in the chimera setting. And we validated these results by treating the mice with an anti-BAF blocking antibody. So actually here, opposite to what we found in with the lymphotoxin treatment, BAF blockade did not affect germinal center formation, but it reduced total B cells in both lymph nodes and spleen. And once again, we found that mutant germinal center B cells were more affected by the blockade of their interactions with the FDCs. And we also evaluated if the FDC network expansion was reflected in primary human follicular lymphomas with mutations in EZH2. So we stained FDCs with the CD21-35 antibody by immunohistochemistry and tissue microarrays that had over 200 FL cases. 17% of them had the point gain of function mutation in EZH2. And we classified the FDC patterns as dense, disrupted, or absent, as shown here in the pictures. And we found that mutant EZH2 FL cases were associated with intact and extensive FDC networks. Now we're in the process of studying the dynamics of the interactions between wild type versus mutant EZH2 B cells with T cells and also with follicular dendritic cells. And for that, what we're doing is intravital imaging with 2 and 3 photon microscopy. This is in collaboration with Chris Xu in Cornell Ithaca. So this is a model that we're using. We're transplanting a mix of bone marrow from tomato reporter crossed with CD4 and then CFP and YFP reporters crossed with C-gamma1 CRE, and one is the wild type, and the other one mutant EZH2. So this image here shows, this is a confocal microscopy image, and it shows two germinal centers in a spleen, where you can see the red cells are the CD4 T cells, the cyan are wild-type GCVs, and the yellow mutant EZH2 GCVs. And the FTC network is here in blue. So here on the left, let's see if I can play this, yes. Here on the left, there is a 3D time-lapse of the popliteal lymph node that was imaged for 1 hour and a half. And the blue cells here are the wild-type GCVs, green the mutant EZH2, and the red are the CD4 T cells. And FDCs here are in white. So here, this is actually, so this is a Z stack, and I'm showing this one because you can more clearly see the follicular dendritic cells. So we can now quantify these images, and not just for the number and the contacts and duration, but also the surface of the cell that is engaged in this contact. And here we can also quantify the trajectory of each cell, the displacement, the length, and the speed. So this 2 and 3 photon imaging is now a work in progress. Okay, so to summarize what we found, we identified some of the molecular mechanisms through which EZH2 mutation disrupt the immune system to initiate malignant transformation. So we have to keep in mind that low-grade follicular lymphomas are tumors that consist of very slowly proliferating lysome germinal center B cells. And given that the centrocytes normally recycle back to the proliferative dark zone, it's actually hard to imagine how these tumors could ever form, because the centrocytes should be overwhelmed very quickly. So our study is providing here the first evidence of how this might happen, where a mutant EZH2 causes centrocyte hyperplasia with expansion of follicular dendritic cells, shutdown of the germinal center recycling from light zone to dark zone, aberrant proliferation of centrocytes and less apoptosis in the light zone, with the clonal dominance and independence of T cell selection. And here is represented by this painting made by my sister Victoria Berlin. So mutations in EZH2 explain the early follicular lymphoma phenotype. And we showed that these phenotypic changes are triggered by profound remodeling of B-cell epigenetic landscape, mainly through this enhanced deposition of K27 trimethylation. But we don't know how EZH2 mutations contribute to the gradual progression of these tumors. So the follicular lymphomas are initially indolent, but they are essentially uncurable. And many cases undergo progression, and they are treated, they relapse, and they become increasingly resistant to therapy. And also many cases also undergo histological transformation to aggressive form of diffuse large B-cell lymphoma. These are the EZV, the LBCL. And these aggressive lymphomas are generally refractory to chemoimmunotherapy. So to understand this process and predict which indolent follicular lymphomas will progress to more aggressive disease, we have started a project now in which we want to determine the epigenetics clonal trajectory through which mutant EZH2 lymphomas gain this progressive fitness advantage during the disease progression. And because the translocations in BCL2 are the genetic hallmark of follicular lymphomas, we have crossed our EZH2 mutant mice with a model where the overexpression of BCL2 is mediated by the excision of this stop cassette in the ROSA26 locus, and is mediated by CRE. And this also occurs specifically in the germinal center stage because we crossed them to C-gamma1 CRE. So we want to identify putative epigenetic configurations that would predict the positive selection of malignant epigenetic clones. So if we can identify these epigenetic clones, we might be able to predict the efficacy of EZH2 inhibitors at specific follicular lymphoma stages. So with this, I want to thank all the people involved in this project, our the collaborators, and also the funding agencies. But especially to Ari Melnick, my mentor. Mentors are always kept as mentors. And then the biocomputational people that analyzed pretty much every sequencing data, Matt Teeter and Shem Maidan. And this is a picture of the whole lab.
Thank you so much, Wendy, for this brilliant overview. I'm not completely sure I can put all the pieces of the puzzle together, but it's really fascinating. The audience can ask questions on the chat box, and I probably will start. Wendy, you put a lot of emphasis on EZH2 mutants in the lymphomagenesis and the consequences on the transformation. But truth is that, for example, in follicular lymphoma, the majority of patients are EZH2 wild type. And the EZH2 inhibitors, such as tazemetostat, that works on both the wild type and mutated, are efficient in follicular lymphoma that are wild type with the same response duration. So what's your explanation? What's the role of EZH2 in the lymphomagenesis outside the mutation?
Right. So this is related to the normal action of EZH2. And so there were many sets of genes that we were actually surprised that were not completely changed by the mutation on EZH2. So there are a subset of genes that are maintained, the same level of repression is maintained in the mutant when you compare to the wild type. So these are sets of genes that we haven't actually explored further, because in this work we focused on those that are more specific to the mutant EZH2. But for example, we think that not the mutant, but actually the wild type EZH2 is keeping the B cells in the germinal center reaction. So this inhibition of differentiation that we thought that the mutant would be like even further repressed, that doesn't happen. So this is the action of the wild type EZH2. So there are many actions of the wild type EZH2 that are really important to maintain the germinal center phenotype. And the EZH2 inhibitors are actually, they are not more efficient in mutant EZH2. They are equally efficient in terms of the enzymatic activity. The EZH2 inhibitors repress both the wild type and mutant. What happens is that the mutant EZH2 cells are more addictive to EZH2. And that's why these cells, or in these patients, are more sensitive to EZH2 inhibitors. But I would say like the bulk of the EZH2 program is actually carried out by the wild type EZH2.
Wendy, just to bounce on your answer and to the question of Frank, do you think that the follicular lymphoma patients that are wild type are not in the same state of the ones that are EZH2 mutant. You said that that EZH2 causes aberrant proliferation of centrocyte, lack of apoptosis, lack of recycling to the centroblast. Would it be that the EZH2 wild type are in a different functional state, or do you think that other mutations compensate and do the same?
No, actually, so unfortunately, we can't compare what lack of EZH2 is doing versus presence of EZH2, because when we remove EZH2, there is not even germinal center. But many of these actions, like interactions with the microenvironment, is dependent on EZH2 itself. So again, what we can study here is the difference between wild type and mutant. But of course, when you inhibit EZH2, and actually we acquired a model where we can induce the deletion of EZH2 in the germinal center, because we have the C-gamma1 ERT model. So in that model, we will be able to address, for example, at the late germinal center stage, if we delete EZH2, what of these functions are required in the wild type set. So again, these comparisons are between wild type and EZH2, but there are many functions of the wild type that also are related to interactions with FTCs and TFH that we are sure are necessary for the progression of lymphoma.
Maybe
we start. Oh, please, Laura, please go.
Well, just to close on this subject, I was wondering, what do you think is going to be then the interaction with the other epigenetic modifiers which counteract the activity of EZH2 or the role of the mutations?
Right. So we have actually we have already crossed the mutant EZH2 with the CREBP and the KMT2D, and of course we see expansion of the germinal centers, and it's so it depends on which mutations we cross to. It's like we obtain different phenotypes. For example, when we cross to KMT2D, it's more expansion of the dark zone. But then we also, so something very important about interaction with the microenvironment is that in the set of just a mutant EZH2 in the germinal centers, we haven't seen any differences in terms of MHC1 and MHC2 expression, which was shown to be downregulated in late diffuse large B-cell lymphoma. When we cross to KMT2D, this is lost. So there are like mechanisms that are, we don't know, and this is something that we are exploring, what's going on at the epigenetic level. So we are now doing all the cut and run to understand how K4 trimethylation, the K27 trimethylation, and also the interplay in the bivalent genes. So we're looking at the mono, di, and trimethylation of K4 to understand what happens to enhancer and the bivalent promoters. But yeah, these are, so unfortunately also in the patients we say mutant versus wild type, but in both of these settings we can have also mutations in KMT2D and CREBP that is difficult to dissociate because there are, like most of the patients with EZH2 mutations, they also carry one or the other. So that's why we're trying to model this and try to understand specifically what happens when we add one or the other. Again, in patients, like I think we got like only 3 cases where we have EZH2 mutations and not many of the other epigenetic modifiers, so we cannot conclude anything.
But so you think it may not simply potentiate the activities, having less activator and more repressor, but also
with the KMT2D, definitely. So we started some lymphoma cohorts with mice, and with KMT2D is definitely more aggressive. Like the onset of the lymphoma is earlier. With the CREBP, it's not that clear.
But also in the absence of BCL2.
No, this is This is
always needed.
Well, yeah. This is always driven by BCL. But I have to say that the like germinal centers, we didn't do lymphoma cohorts, but germinal centers with the double mutations KMT2D and EZH2, are like 10-fold increase. So they might be able to generate lymphomas. Yeah.
And can you tell us more on the effect of EZH2, either mutated or wild type, on the plasticity of T cells, but not only T follicular helper cells, but also effector cells and others that could be involved in combination, for example, with IMiDs, to see whether there is a kind of additive or synergistic effect.
Right. So we have done some work in collaboration with the Christian Scale Lab. And so they were looking at infiltration of CD8 and CD4 in already established mutant EZH2 lymphomas with the BCL2 translocations. And when we treated the mice with EZH2 inhibitors, we saw more recruitment of CD4 and CD8. So we still don't know the mechanism of the mutant EZH2 in already set lymphomas. This is something that we're studying right now. But this is what we observe when we treat with EZH2 inhibitors. So somehow it looks like the mutant EZH2 is avoiding the recruitment of CD8 and CD4 cells. So it looks like the inhibition of EZH2 is reestablishing this contact. So maybe what we observed here, this is like the basic mechanism of how this occurs. There are also, of course, indirect effects, like for example, the EZH2 inhibitors also induced overexpression or downregulation of certain cytokines that are involved in recruitment of macrophages, natural killers, and other subsets. So we're now in the process of trying to understand and dissect all these mechanisms.
Questions from the audience. Do you have clues on how EZH2 may impact on NF-kappa B activity in follicular dendritic cells, since EZH2 impacts BAF, LTB, and TNF?
No, this is something that I'm trying to study now. So, so the interdependency between them is kind of obvious, but we still have to formally confirm this, because yes, FDCs will need TNF and lymphotoxin for their expansion, and we saw that indeed mutant EZH2 are overexpressing these molecules, and then, like by the same token, mutant EZH2 B cells will need BAF and survival factor from follicular dendritic cells. So far we haven't looked specifically on follicular dendritic cells, but this is work that we are doing in collaboration with Karin Tart that you might be here in this session. So we are studying all the, not just FTCs, but all the stromal subsets in the context of mutant EZH2 lymphomas.
Another question from François Lemonnier. Do you think that EZH2 inhibition restores the T cell dependency of FLB cells?
Well, yes, this is what we observed. So again, in advanced stages, the data we have is just by treating the cells or the mice with the inhibitors, and we see, the reestablishment of this recruitment. But these are experiments that we are actually planning now. So far, again, all the data I show is like a pre-neoplastic stage of germinal center B cells, and we are trying to understand this mechanism in more advanced stages. And we know that the mechanism may change over time. Like as I just talked with Laura about the MHC1 and MHC2 expression that is not changed at the germinal center stage, like pre-neoplastic stage, and they are downregulated in advanced DLBCL, in the mutant EZH2 specifically. So this mechanism might change over time, and this is something that so what we're doing is we set cohorts of mice and we are euthanizing at different time points to try to understand this mechanism along with this epigenetic clonal evolution that I talked about at the end.
But do you know if, for instance, in the mouse tumors that develop in this model, there are genetic lesions that are acquired in the MHC class 1 genes, for instance? Like
yeah, we don't know. This is something that we're collecting all these tissues because that could happen. It could be. So from some of these mouse models, we're also trying to generate cell lines, and we have to sequence them because some of them became really aggressive, especially when they are able to be cultured ex vivo, and then they are actually like cell lines because they survive forever. And I'm pretty sure they have acquired other mutations, maybe MYC, p53, or yeah. So we have to sequence all, not only in the cell lines that we are trying to generate, but also in the different stages to understand what if there are other genetic lesions that are. So I know in the context of the ROSA26 PCL2, together with the ROSA26 MYC from Bruno Amati work, they have found that late stages acquire some genetic alterations. So it's not just driven by this. This is something we have to.
Yeah, well, but otherwise the whole mouse would become a tumor, right?
Right. Yeah.
There would be some. That's very interesting.
What do you think of long-term epigenetic therapy with all the knowledge you have on epigenetics? Do you think it makes sense from the clinical perspective? Aren't we going to have trouble in treatments given for more than 3 years?
Right. So of course, I think it doesn't make any sense. And because we have some preliminary data of these mice, I was telling that we are collaborating with Christian Stadel, that we have actually treated mice. It's always in the context of mutant TCH2 BCL2 because they were very sensitive to the inhibitors. So we treated the mice only for one cycle. One cycle is 28 days, same as in patients. We stopped the treatment, and we euthanized mice right after stopping the treatment, and we see that K27 trimethyl is really down, and everything as expected. The tumors are more regressed in the EZH2 inhibitor treated. But then 3 months later, after finishing the treatment, so this was only 28 days treatment. 3 months later, we euthanized mice. We looked at K27 trimethylation, and it's still repressed in the mutant TCH2. So this might mean that the patient don't really need to be treated for so long. But I have to say, this was a pilot experiment. We treated very few mice, and this is something that we are trying to reproduce also with other genetic alterations, like MYC, for example.
But apart from that, what could be the negative consequences of a long-term treatment on the other cells of the microenvironment or on the myeloid cells, or what would you think?
So the effect of EZH2 inhibitors, what have been shown in patients is in other cells. So it depends on the cells. So for example, in the Tregs, they are actually it's beneficial, the EZH2 treatment, because they have the FOXP3 is also regulated. So EZH2 is actually necessary to stabilize FOXP3. So when you treat them with EZH2 inhibitors, you are inhibiting Tregs. So this would be beneficial because it will attract the other effector T cell population. But there are other cell subtypes that they have an advantage after EZH2 treatment that would be beneficial for tumor growth. So I don't know how this would pan out when you take it like globally, because it has effects in many. all these kind of cells that are expressing EZH2, of course, they will also be affected by EZH2 inhibitors. So what have been seen in patients is that usually is beneficial. My prediction for very long-term treatment, in terms of resistance, we haven't actually observed that. So at least in the context of the mutant EZH2, we actually tried to generate resistance cells and we couldn't. And I don't think that would be like a major problem, like generating resistance cells, but it will be more in terms of the microenvironment. And again, so there are so many players involved that on one hand will benefit the tumor, and on one hand will, on the other hand, will decrease tumor progression. So I don't have the answer for this. We'll have to do the experiment in mice for short and long terms and see.
But
there are companies.
Sorry. Can I just bounce on this? For acute leukemia, loss of function of EZH2 is causing acute leukemia.
Yes, of course. Yeah.
But this has not been observed so far, or?
Exactly. Yeah. So it's a totally different role. Maybe because of the targets of EZH2 in leukemia that are different than in lymphoma. But as far as I know, the EZH2 inhibitors were not inducing leukemia, as far as I know.
Many patients have been treated, heavily pretreated before. So you have cases of myeloid leukemia, myelodysplastic syndromes, those patients. And we recently had one case of B-cell acute lymphoblastic leukemia in a patient that was EZH2 muted, but not on the leukemia, but on the follicular lymphoma. Do you think this patient had received 4 years of therapy? Do you think the treatment could have an impact in the generation of the acute lymphoblastic leukemia, which from the cytogenetic perspective really looked like an induced leukemia?
So this patient received 4 years of EZH2 inhibitor therapy. This is what you're saying? Yeah. Okay.
But before chemotherapy and 2 or 3 lines of chemotherapy before.
Right. So it's reasonable that it could. It's actually reasonable because of the need of EZH2 to refrain this leukemia. But I don't know. I mean, we would have to experimentally test this. I think it's reasonable. Yeah.
And one question from Bertrand: there are companies developing dual inhibitors targeting EZH1 and EZH2. What's the value of such a drug from your perspective?
So EZH1 is actually, so when we looked at the levels of EZH1 in germinal center B cells are not that high, but they are actually expressed, and they are expressed in diffuse large B-cell lymphomas. So we think that EZH1 might be necessary for maintaining the K27 trimethylation mark. So we are now also working with the dual inhibitors and the single EZH2 inhibitors to try to compare, especially in the context of the immune synapse and how the immune niche is changed by these inhibitors, because we know the molecular actions, but we don't know what happens with the immune synapse. But the dual inhibitors might add like beneficial therapy. This is something that is a hypothesis, but these are experiments that we are planning to compare both.
So you mean for diffuse large B-cell lymphoma, but this shouldn't be the case in follicular lymphoma, or it could be also to a lesser extent?
No, it could be. No, I think it could be. It's like a progression. So I think it should be also valid for follicular lymphomas. We have this data from DLBCL, but I'm sure it will be the case also for FLs. Yeah.
One more technical question from Raima Zian. Do you look to RNA pol 2 occupancy in your conditions?
We haven't done that. We are actually doing now. We are looking at the early transcript, not pol 2, but we're looking at the early transcript by ProSeq. So this is data that we're cooking.
One more.
Sorry, go ahead.
One question.
Got another question related to your vision of your data of showing that EZH2 mutant is causing proliferating centrocytes. Do you envision that those centrocytes are stuck as centrocytes like the ones that are proliferating, and they are different from centrocytes? But do you think that they are stuck in that state, or they are still very dynamic, such as normal B cells?
So in the context of only mutant EZH2, we think that they are stuck at this stage, but when you look at the germinal centers after a long time, they are also resolved. So they are not actually, they are temporarily stuck in the light zone, but after a long time they are eventually resolved. So this means that you actually need a second hit to keep this light zone phenotype. And we know that, of course, when you combine with BCL2 mutations, this super expansion of the follicular dendritic network and the light zone phenotype is maintained, and it's actually progressing over time. But the mutant EZH2, yeah, so it's definitely keeping them. They are not, again, recycling because of, the clonality and the MIG reporter that we have done. But they are not totally stuck.
Okay, so that would reconcile with our own data. I had trouble to reconcile that vision with the one that we're starting to have with single cell analysis. That those cells in follicular lymphoma, at least, are, without knowing if they are EZH2 mutated or not, but in general are very dynamic and can transit from a germinal center-like to memory-like, sometimes to PC-like, and go back to GC-like. So I'm glad to hear from you that your vision is not that they're completely stuck.
Well, we have actually done, this is very, very recent. I think I had the results only last week. So we have done memory recall experiments with the mutant EZH2. So in the first setting, so if you take B18 cells with mutant EZH2 and you see them in wild-type mice, the percent of memory B cells coming from the mutant EZH2 is much bigger than the wild type. But when you take those cells and put them, after several months, you take the memory B cells, NP positive, and you transplant them into mu-MT mice, we have much bigger germinal centers in the mutant EZH2 than in the wild type. So they are definitely becoming memory B cells, and they are able to reseed germinal centers. Yeah, okay. They are more dynamic in that. Yeah.
We agree. Great.
What do you think of? What are, to your opinion, the most other interesting drug candidates or targets in terms of epigenetics? For example, what do you think of KDM5 inhibitors?
So, not as a let's see, in the context of wild type, so mutant EZH2, but wild type for other epigenetic modifiers, I'm not sure if that would be something beneficial. But in the context of mutations in other epigenetic modifiers like KMT2D and CREBP, the combination therapy could be beneficial, but I honestly, I think that the most beneficial therapy would be like something in the microenvironment rather than other epigenetic modifiers, because of all the effects that we see that is altering in this immune synapse. We think that the more beneficial therapy would be targeting another level, not another. But again, this is something that we should actually test.
So do you think it would be reasonable to combine EZH inhibitors with bispecifics, antibodies,
right,
T-cell engagers?
Right. Yeah. So this is our next step, immune therapy, exactly. And of course, here at Cornell, we are collaborating very closely with Roberta Sapasodi, which is an expert in immune therapy, and she's exploring this immunotherapy aspect in the mouse models that I generated, and I will be focusing more in the in trying to target FTCs. Like, for example, with BAF blocking antibodies. And, yeah.
Okay. Any more questions from the audience, or?
Maybe I ask you something which is probably naive at this point, but so what do you think about the combination between instead EZH2 inhibitors and checkpoint inhibitors, which may have some counter aspects?
So I think it actually depends at the stage of the of the tumor, because it's as I said before, it's going to be a totally different immune niche at, for example, early FLs or advanced or transform FLs or aggressive DLBCLs. So I think it really depends on the stage, and that's why we're also trying to understand the how these cells are behaving at different stages, and not treat them as a unique, like the same kind of lymphoma. It's actually, at least what I have observed at the pre-neoplastic stage, and compared to our models when it's already transformed, I think it's like totally different scenario. Again, it could be that, as you mentioned before, there are other genetic modifications, but I think that each case has to be considered like individually.
Because in view of also what you said, right, the effect of the EZH2 inhibition on Tregs.
Yes. So actually, so this would be beneficial, but then on the other hand, it can also affect CD4 helper cells, and then others. So there are good and bad parts about the EZH2 inhibition. For another one that we observed, it's a very preliminary data, but it looks like EZH2 inhibition is affecting macrophages, but in a good way for the patient. So it looks like the polarization is more towards M1. But in combination with immune we will, again, it will depend on the stage. And I'm talking specifically about the combination with EZH2. So other epigenetic modifiers might be a totally different scenario, like KMT2D and CREBP. So
it is complicated.
Yes, it's not that easy.
And combination of EZH2 inhibitors and BCL2 inhibitors are in the air for more than 5 years, but there is no clinical data and nothing has been launched so far.
Yeah.
Do you think it's realistic, or do you think it's just nice in mice?
No, I think so. Here at Cornell, with Dr. John Leonard, they are so they launched already a clinical trial to combine venetoclax with tazemetostat, based on the work from also here from Cornell, Lisa Roth, in PDX, DLBCL. The combination like really killed the PDX. It was like amazing. So based on that, they are launching this clinical trial. So I think this is something that is actually happening. And in preclinical trials, I will be also testing venetoclax tazemetostat with BAF inhibition, BAF antibody, blocking BAF antibodies. But yeah, no, I think this is going to happen sooner or later.
I'm sorry, but I think we're gonna have to close the session unless there's one last burning question, Frank and Laura.
No, I think Wendy did a great job. We learned a lot.
Brilliant talk, brilliant session. Many thousand thanks, Wendy, to fill in for Ari. We even don't need Ari anymore. I'll tell you.
Don't tell him.
It's done for us to send brilliant speakers instead of oneself. Thank you very much, very much. This was the last Red Talk of the season, brilliant one. I wish you a very nice summer holidays for those who will have one. It's not still yet, but we will see you back in September for another Red Talk session, which might be announced very soon. Thank you very much to all. Have a good day or night, and see you very soon.
Thank you.
Thank you. Goodbye.
Bye-bye.